Good morning, everyone. Welcome to the Reliable Classes. Today, we are going to study Chapter 2 Cell, the Unit of Life of Class 9 ICSE. So, before we begin, let's see what is cell. Cell is the fundamental, structural, and functional unit of all living beings, and the study of plant and animal cell is called cytology. You have studied about cell in the lower classes, and let's recall that it is a fundamental structural and functional unit of all living beings. Let's move to the next slide and let's see more facts about the cell. It is the smallest part of an organism which is capable of independent existence and of performing the essential functions. And every organ of our body is composed of hundreds of thousands of cells, whether it is your brain, skin, or your muscles, or even bones. These are composed of cell. And even similarly, if we compare to the plant, even leaf, wood, roots, everything is composed of cell. And every cell has its own life. Old and weak cells are replaced by new cells. As you might be knowing, your RBCs or red blood cells, for instance, die after about 120 days. And these cells are really very small. You cannot see them with your naked eyes. You need a very complex device to see them, and that's a microscope. And the most important thing, that most organisms, including ourselves, start a life as a single cell called zygote. Let's see about microscope. As I told you in the previous slide, the cells are very small and cannot be seen through a naked eye, so they can be seen under microscopes. And there are three kinds of microscope: simple, compound, and electron microscope. So let's see the discovery of the microscope. So the first microscope was constructed by Antony van Leeuwenhoek. He was Dutch scientist, an ordinary public official who ground lenses and made a microscopic observation as a hobby. And it is said that he had constructed 400 microscopes. Basically, the microscopes which were constructed by Antony van Leeuwenhoek were the simple microscope. It consists of single biconvex lenses and and how to see an object through the microscope made by Antony van Leeuwenhoek. See, this is the front view. Here are the lenses. Here is the focus knob. Here the sample holder. This is front view and this is the back view. Where you can see B is the back view and the plain one is the front view. So on this side, the eye was kept closed and the object was mounted on this sample holder here. This is a needle-like screw on the opposite to the lens, and through this we can see the object. We have to mount it on this sample holder. So let's move on to the next slide and see another discovery of microscope, which was done by Robert Hooke. So Robert Hooke, he developed a microscope using two lenses of greater magnification, and such a microscope is called compound microscopes. Robert Hooke made a compound microscope. Here the microscope which is made by Robert Hooke. So this is the stage where the object is kept. and a light from the oil lamp was thrown on it. Here's the oil lamp, and the light from it was thrown to the of this stage, and by the means of concave mirrors. Keep this in mind. Robert Hooke used concave mirror in his microscope, and he observed different things under microscope. And when he saw a cork under a microscope, when he saw cork under a microscope, he was amazed. He saw a box-like structures which recalled him of a place, or reminded him of the rooms or cells of monks in a monastery. So he said that cork was made up of cells. So ultimately, the discovery of the cell was done by Robert Hooke, but he saw the dead cells. The cells which he saw were dead and had only empty boxes or walls. Let's move on to the slide. So, as you know, the compound microscope which we use today is the improved version of the Hooke's microscope. Okay, compound microscopes are used to view small samples that cannot be identified with our naked eyes. These samples are typically placed on slide under the microscope, and it uses light which is bent by a glass lens to magnify. So there he uses an oil lamp, and we use a light which is and we are using glass lenses to bend the light and to magnify the image. But he has used an oil lamp and a concave mirror to magnify the image, and the magnification of the compound microscope is 2000 times, and that of the simple microscope which was made by Anthony van Leeuwenhoek, that the magnification power of that was up to 200 times. And here let's start with electron microscope. It is most sophisticated microscope, can give magnification to up to 2000 times, and this one uses a beam of electrons which are bent by the magnets. An electron microscope can be used to explore the molecular nature and mechanism of diseases, to view the 3D structure of biological tissues or cells, determine the structure of proteins, and observe viruses in a biological context. So let's see about the cell theory. So the cell theory was proposed by Matthias Schleiden and Theodor Schaben. So first, Matthias Schleiden in 1838, he was a German botanist. He said that every plant is made up of a large number of cells. Then 1 year later, Theodor Schaben, a German zoologist, suggested that all plants and animals are composed of cells which serve as the unit of structure and function. So they together proposed a cell theory in 1839. Then later on, Rudolf Virchow, he made his addition to the cell theory in 1858, that by saying that all cells arise from pre-existing cells. So here is the cell theory. So first, cell is smallest unit of structure of all living things. It is the smallest unit of structure of all living things. Then cell is the unit of function of all living things. and all cells arise from pre-existing cells. So if we examine any part of body under the microscope, we should show a cellular structure. It will show a cellular structure. And if we see the second one, the unit, any function in the cell body of all or living organism is due to the activity of cells, like example of movement, digestion, photosynthesis, etc. And third one, all cells arise from pre-existing cells. This means that cells die and are continuously replaced by the new. Most life starts from a single cell. And for the multi-celled, we have human beings and few plants. Most of the plants and human beings or animals we can see, like example human beings, mango trees, and few cells are like Spirogyra, volvox. I mean, there are only relatively fewer number of cells, it's just few hundred or few thousands. And most of the plants, animals we see around us are made up of millions and billions of cells. So here we came to the end of the session of the video. So thank you everyone. Do like, share and subscribe our video, and do comment, and don't forget to click the bell icon. Thank you everyone.